Hello, welcome all to the webinar Connecting Adverse Outcome Pathways, Knowledge and Data with AOP-Link Workflows. I'm Marvin Martens. I'm very excited to present to you today about our case study in OpenRiskNet. Before starting, I would like to remind you about all the previous webinars, past events that have taken place, of which web recordings are available through YouTube and on the OpenRiskNet website. Also a reminder for the upcoming final workshop, the OpenRiskNet final face-to-face workshop taking place in Amsterdam on the 23rd and 24th of October this year. So to kick off this webinar, I would like to briefly describe the concept of adverse outcome pathways. Of course, I am pretty sure many of you know the concept, but to the ones who are not familiar with the concept, I will briefly explain what they are and why they are very useful. So basically, we can envision the adverse outcome pathway concept as a bridge between scientists, who create toxicological data, experimental data, and the risk assessors, the regulators, to perform knowledge-based regulatory risk assessment. AOPs are basically a chain of biological events. They capture mechanistic toxicological knowledge of biological processes, toxicological processes, to support decision making. In the figure, you can see a brief schema of what is an adverse outcome pathway, which is initiated with a stressor causing a molecular initiating event, leading to a cascade of events called key events, which are biological responses that follow the initiating event and lead to the adverse outcome. All of these events are measurable and essential for progression and cover all levels of biology, in which they start with the molecular interaction at the molecular level, and through cellular responses, tissue responses, and organ responses lead to the adverse outcome. How do AOPs fit in the risk assessment? Basically, they aid integrated approaches to testing and assessment. It's for short IATAs. They allow the reuse of information, the reuse of knowledge, the reuse of data. And this leads to more effective, more quicker and cost-efficient risk assessment. They have predictive value. They reduce the amount of animal testing that is necessary to have a valuable risk assessment. And they are used to identify knowledge gaps because they capture all of the information that is already available. They can also show where there is lacking information or lacking data to support the adverse outcome pathway. This brings me to the core topic of this webinar, which is the OpenRiskNet case study called AOP-Link. Or what is actually the AOP-Link case study? Basically, we are trying to answer two main research questions. We want to make the connection between experimental data and adverse outcome pathways. So our first question to answer is, for a certain AOP, which experimental data is available to support the AOP? And the other way around, can we support an existing AOP through this experimental data? Can this experimental data support an existing AOP? So basically, AOP-Link is really the link between AOPs and experimental data. If we look at the broader overview of the hazard assessment framework, which was developed in the SURAT-1 project some years ago, we can see that the AOP-Link actually covers two main components of this whole framework. In Tier 0, Step 3, it can be used for collecting supporting data, of course, based on the adverse outcome pathways. And in the Tier 1, Step 6, mode of action hypothesis generation, also by using adverse outcome pathways and the data that can be connected to it. So the main component of this case study is the AOP-Wiki. It is a central repository for adverse outcome pathways, qualitative adverse outcome pathways, and is part of the larger AOP knowledge base, which is a joint effort between the European Commission Joint Research Centre and the US EPA. Currently, the Adverse Outcome Pathway Wiki, AOP-Wiki, contains over 3,000 entries covering adverse outcome pathways, key event relationships, and stressors as their main components. If we enter the AOP-Wiki, we come up with pages like these. On the left side, you see the initial overview, what you will see if you go onto the AOP page, listing all of the AOPs in a big table. If you click on that, you go to a more detailed overview, in which you can see the title, the contributors, the authors, the graphical representation, and all of the information about it below, which you can see on the right side, for example, the domain of applicability, and all of the assessments that are part of the adverse outcome pathway. And the same counts for key events, stressors, and key event relationships. However, with this information, all of the contents, all of the information is covered inside these large pages. So to go a little bit more in detail, we go into one of the AOPs, which is the protein alkylation to liver fibrosis adverse outcome pathway. You will see this one occurring more often in this presentation. So basically, an adverse outcome pathway consists of one main page to describe what is the adverse outcome pathway, describing the abstract, the background to which it is applicable, and what components it has, and the overall assessment, of course. Next, there's the stressors that link to the molecular initiating event, which are often chemicals, but can be other things as well, and has some information about evidence and description of the stressor. Then there's the key events, which by themselves are separate entries in the AOP-Wiki and contain by themselves a lot of information all stored in one large page. And the same counts for key event relationships, all covering a variety of types of information, both structured and unstructured free text, but all the same, not accessible, not queryable for more specific. So the main issue with the AOP-Wiki that it mostly contains free text, tables, and some identifiers. And for the AOP-Link to work well, we would like to make this database, the AOP-Wiki, more accessible. We want to extract specific information to retrieve specific answers that we have. Also, we would like to see harmonization with other databases. By using persistent identifiers and ontologies, linking databases should become better. We call this interoperability. The solution for that is semantic modeling of the databases, in which we annotate the relationships with ontologies and entities with persistent identifiers. To do these semantic annotations, we chose to develop the AOP-Wiki RDF. Briefly describing RDF, standing for Resource Description Framework, it is a standard to describe information in web resources, and all information is captured in this system of triples, stored in triples. As seen in this figure, consists of a subject being the main component that you describe, predicate being the relationship towards an object, which is the property of the subject basically. This RDF is generally used in databases, and there's a large number of libraries and tools that use this kind of framework. So one of the main goals in AOP-Link was the conversion to RDF, to go from the initial full page descriptions that we see on the left towards a more structured, detailed overview as seen on the right side. But we do this not so fast. We do this step by step. The existing AOP-Wiki consists of these 4 major components, which were shown in the earlier statistics table, being the adverse outcome pathways, key events, stressors, and key event relationships. However, if we look in the underlying database, we can see there's much more components that are very important to link to other databases to find the ontologies and the identifiers. For example, with taxonomy or chemicals, but also the ontological annotations of cell terms, organ terms, and the key event components, which consist of the biological process, object, and action. And all of these components, all of these objects are connected through identifiers. Some of them internal, so invisible to the public, some of them with public identifiers, which are registered in as persistent identifiers as well. And by annotating all of the relationships that these properties have, by describing all of the predicates that occur in this schema, we can know in more detail what is the meaning really of the connection, what is really the meaning of the predicate. For example, if we have an adverse outcome pathway and we would like to know what is the molecular initiating event of this adverse outcome pathway, there is a relationship property that has that uses the AOP ontology as molecular initiating event. Things like this make using the RDF much simpler and straightforward, and also aligns with other tools that use the AOP ontology, but also other kinds of RDF systems, databases. So to go back to the simplified version with the 11 main components, we can elaborate on this by adding all of the properties that are captured inside of the AOP-Wiki. And then you end up with something like this, in which all of the 11 components have a variety of different components. And modeling the database in this way allows for querying for singular components, singular properties of every object. So you can query the database in much more detail than it was before. How to really use the RDF that we developed? There's basically two ways in which we expose the AOP-Wiki RDF. The first one is through a public SPARQL endpoint, which is currently not working, but will soon be. So that's why I kept the link there. It is basically a virtuoso SPARQL endpoint in which the data is loaded. And if you know a SPARQL language, which is the query language to query RDF, one can query any type of information and make all the connections that they want from the database, as long as you know the structure of the data. The other component that is used to access the data is a REST API that we developed with using grlc, a tool that develops an API on top of SPARQL queries. So in this API, SPARQL queries are loaded with parameters that are preloaded and predefined. So it's very easy to use. Of course, this is limited as you only have a preloaded set of queries. So I want to show a few examples of what we can do with the AOP-Wiki RDF. So the first example here is for AOP38, which is the AOP that we saw before. As you see on the bottom, the protein alkylation to liver fibrosis. We would like to have all the key events, the identifiers of the key events, their names, and the measurement methods, which are separate components of the key events that are part of this adverse outcome pathway. And we can do this in basically two ways. On the left side, again, the SPARQL endpoint, we can add the query, which is a fairly simple query for retrieving the methods, where we filter for the adverse outcome pathway number 38. On the right side, there's the Open API, which has the preloaded query in there and the parameter value. So on the bottom right, we can see that we can fill in a number there, which is the AOP that we are interested in. If we fill in there 38 and drag up and press on execute, we can actually we actually get the same result as we would get in the SPARQL endpoint. And that would look like this. In general, SPARQL endpoint spits out tables in the way that you like it, in the way that you request. So for this query, we requested only the key event identifier, the key event name, and the text that is captured in the method description. And the outcome for both of the tools that I described look the same, look like this. How can I go next slide? Yes. Another example, which is currently not possible with the existing AOP-Wiki database or AOP-Wiki website, is searching for AOP-related components based on chemicals, based on chemical identifiers. In this example, we query for the CAS registry number 107.18.6, which is allyl alcohol, which is one of the stressors that was one of the chemicals linked to stressors that causes the AOP that I presented before, the protein alkylation leading to liver fibrosis. We are now able to query in one single query for all of the AOPs that have this stressor, this chemical related to the stressor. And this is a really powerful way of extracting information, because this is not possible to do in one single way in the AOP-Wiki. One has to find the stressor, one has to find all the pages that are related to it, and read all of the information about the chemicals. The same goes for the second example on this slide, in which we do not just search for one single identifier, but we can extend our search. We can extend our query to have basically unlimited amount of chemicals that we would like to ask, that we would like to find the AOPs for. And this is a very powerful tool, because you can extract information from a lot of different pieces of information all at once, and ask for many questions all at once. Another thing that we added to the RDF is with BridgeDB. By using BridgeDB, we added additional identifiers for chemicals mostly to the AOP-Wiki RDF. For example, for CAS registry number that we talked about before, allyl alcohol, we have the identifiers now of other databases such as ChEBI, HMDB, ChemSpider, and Wikidata. And this allows for many more connections to external databases. If one does not have the CAS registry number, which is currently the only identifier system that is used in the AOP-Wiki apart from the Comtox dashboard ID, one has no other option to query the database based on the chemical identifier. But now this is possible. So this example covers that, in which we use the ChEBI identifier, also the one for allyl alcohol, which we saw in the previous query, to query for all of the adverse outcome pathways that are related to this ChEBI identifier, this chemical. And this is, again, something that is absolutely not possible without parsing the complete database and mapping identifiers. But now it's included inside the RDF, and we are loading this kind of queries inside of the user interface as well, the REST API. The last example, fourth example that I like to give, is the query to answer, is the query for all chemical names that lead to a specific adverse outcome. So making a long distance query from an adverse outcome of an AOP, we query of which adverse outcome pathway it is a part and which molecular initiating event it has. From there, we want to know which stressor relates to this molecular initiating event and which chemical is related to that. So this kind of question also covers many AOPs all at once. As we can see in the first example, we queried for all chemicals that lead to learning and memory impairment. And we see that there's at least 10 chemicals or 9 that lead to this, this adverse outcome through different stressors and through different adverse outcome pathways. Also, the other way around, we can ask. Also, the other way around, we can ask for the database to give all of the adverse outcome pathways based on a certain molecular initiating event, based on the activation of a particular molecular initiating event. In this case, AHR receptor activation, in which we see that there's at least 3 adverse outcomes that can be the result or that are linked in the AOP-Wiki to this molecular initiating event. As shown before, there's the 2 ways of querying the database, of using the RDF. But we also presented grlc, which is a tool that builds the API on top of your SPARQL queries. And basically the advantage of using this tool is that there's no need to learn the SPARQL query language. As shown in the first little figure, that is the figure that we used for extracting all of the methods for a particular AOP, in this case number 38. The benefit of using this tool, this grlc, we can use the interface and enter parameters. So we can reuse the same query for all AOPs that we like, for all we can fill in basically all identifiers that we want, which makes it very modular as well. Also, it allows for direct requests from the command line, but also through a URL query string. For example, the link that is seen on the bottom, one can directly retrieve the full table that was seen before with all the methods for AOP number 38. Unfortunately, again, this tool is currently not working due to the SPARQL endpoint not working, but that will definitely be solved very soon. However, the page is available and all the queries are there. And if you have any requests for specific queries or specific questions to be answered or to be added to this interface, do not hesitate to go on the GitHub page and ask for it, to file an issue and ask for additional queries. I will be very happy to make them. The next section is about integrating the AOP-Wiki RDF in a larger workflow. Instead of just using the AOP-Wiki as itself to extract information and link it to other databases, we would like to see what more it is capable of by using Jupyter. So I will show one example instead of what I mentioned in the abstract multiple due to lack of time, for which the main question in this example is, does the AOP-Wiki contain AOPs relevant to nanomaterial toxicity? The approach that we took is extracting first all chemicals from the AOP-Wiki, of course using the SPARQL endpoint, and then using the ChEBI ontology to see if they are related to ontological annotations of the nanostructure. Another approach after that is actual direct text matching of AOPs, key events, and stressors to see if there's any related to nanomaterial toxicity. The result is that no nanomaterials were found through the chemical identifiers, but based on text matching of AOPs, key events, and stressors, we found that at least 10 AOPs are linked to nanomaterial toxicity. I'll briefly go now through the notebook that we prepared for today, which I developed together with Serena from my group. So after the first setup steps, we start using the AOP-Wiki RDF. We extract using SPARQL wrapper in a Python notebook. We create this SPARQL query, which is also present in the API if you are interested, to request all chemicals of the AOP-Wiki and basically put them in a big list, in a big table. Related them to the adverse outcome pathway that they are part of, and of course the CAS register number. We would like to have the identifier After this, we want to connect this to the ChEBI ontology. So we would like to have the ChEBI identifier for these chemicals as well. So what we did after that is use the BridgeDB tool to map all of the CAS registry numbers to ChEBI identifiers to be able to query these chemicals in the ChEBI ontology. And put this in a table. Of course, this required some cleanup. We removed some empty rows, and we ended up with about 500 ChEBI identifiers that we would like to look up. Then the next part was to ask the ChEBI ontology for all of the terms related to nanomaterial, nanostructure, I should say correctly. And we extracted all of the child ontology IDs of nanostructure and put these in a large list, ending up with about 124 ontology annotations. Then looking through all the chemicals, extracting all of the ontological annotations of the chemicals in ChEBI, and after matching this to the set that we created with the nanostructure search, we found out that there is 0 intersecting chemicals with the right ontology terms that we predefined, which are basically related to nanostructures. The next thing that we did was then the search for text, the text mapping search, because we found no connections through the real identifier annotations. We were still curious why we didn't find anything, because we knew that there's a few projects going on that are developing nano-related AOPs. So again, using the SPARQL queries, shown here in red, we queried for all of the AOPs which have in their description the word nano. Of course, this doesn't say much yet, but also we try to extract the full word of what is this word nano coming from, this part of nano. And basically this gives us 3 AOPs in which we find in the description the term nano as a part of larger words, being nanomaterials, nanoparticles, and a multitude of nanoparticles. So only 3 AOPs are found directly through chemical or through text mapping of the description. The next thing that we did was going into the key events, because they often contain more biological information about the events going on. So their description contained more of the biology aspect of the adverse outcome pathway. And this actually led to a list of 7 key events that we can, in which the word nano is contained as part of nanomaterials, nanotubes, nanoparticles, things like this. The third thing that we did, the third query that we performed, was searching for the word nano inside the titles of stressors. And this thing gave us the most results. It gave us 13 stressors, 13 AOP stressors that has the name nano in its title, as presented here in this table. In the second column, basically, you can see all of these stressors that are part of different AOPs that contain nano in their name. You see the nanoparticles, the silver nanoparticles, the silica nanoparticles, carbon, etc., nanotubes. And we see also on the right side that none of these stressors have a chemical related to them. So there's no annotation of chemicals for these things. And this is a real big issue when working with nanomaterials, but that's a whole different story. So collecting all of this data and gathering everything together, we find that in total, there are 10 AOPs related to nanomaterials and 8 AOPs related to 8 key events related to nanomaterials through the key events themselves, but also through the stressors. And that's basically the answer to the questions that we had. Does the AOP-Wiki have adverse outcome pathways related to nanomaterial toxicity. And although we cannot find the answer directly, we can do it through 3 different searches. We were able to define 10 different AOPs that are occurring multiple times based on the query that have this word nano in and have a high chance of being involved in nanoparticle toxicity. Let's get back to the slides. So this shows you a brief story of how we can use the AOP-Wiki RDF. Of course, there are many more examples how to use this. And I would like to ask you, what is in your eyes a good way to use the AOP-Wiki RDF and connect it to external databases? So I asked Lucien to set up the poll that is present in the screen right now to ask which kind of data or knowledge would be beneficial to adverse outcome pathways that should be integrated in Jupyter notebooks that is not yet presented. Of course, we covered a little bit of the chemical identifiers, the chemical databases, but there are a couple of others. So I'm curious what the audience that is present in this group, in this webinar, thinks about this. I will give you a few moments. We will, for example, be curious how adverse outcome pathways, or at least the adverse outcomes, relate to disease databases. Can we make this connection? Or population genetics, are certain groups of people more susceptible to a certain toxicity of a chemical or nanoparticle? And also molecular pathway database, which is one of the main topics of the AOP database as well. Is the poll done?
Yes, you can see the results, right, on your screen?
No, I don't see. I still have my own slides in the view.
I see. So basically you have 60% for the first answer for the diseases, and 30% for the chemicals, 20 for gene variants, 20 for population, and 50% for the last one, for the molecular pathways.
Okay, great. Ah, that's nice to see. Really a majority on the disease databases, phenotype databases. That's really interesting to see. And indeed, I think that the AOPs, they cover a lot of data, a lot of different levels of biology, and they lack a bit of information on the higher level of biology, the phenotypes, the population level key events. So that is indeed something that is very interesting to look into. Thank you. I was curious what people would say. This is a good result. Oh, so for the final bits of the webinar, we are going to look how AOP-Link fits in a broader aspect, in a broader perspective. Because AOP-Link is part of the OpenRiskNet project, it is one of the 7 case studies. We can actually connect it to other case studies that are in the project. So these 3 on the left side, you see 3 case studies called DataCure, TGX, and SysGroup, which actually have direct connection or can have direct connections to AOP-Wiki, to the AOP-Link, sorry. Indirectly or directly. On the right side, we have a more detailed overview in which we have all the tools, databases, and some of the steps that happen inside of these case studies, in which purple indicates the TGX case study, yellow the DataCure case study, blue the SysGroup case study, and AOP-Link on the bottom, the green, the green section. And we can see that if we have this connection between the different case studies, we can actually see quite a lot of nice paths of tools happening. For example, if we have a certain data set, for example, through DIXA, there's TGX that can make predictions. They make predictions. Their goal is to make prediction about the activation of certain molecular initiating events. And this prediction model can then be linked to the adverse outcome pathway wiki, see if this molecular initiating event is there, what kind of AOP is it connected to, what kind of methods do I need to test this. But also we can go further downstream using the AOP database RDF, which will also be there in the future, or the WikiPathways RDF, which is already present in the AOP in the OpenRiskNet infrastructure. On the other side, we can also use the data that comes from DataCure to group chemicals based on chemical similarity and mode of action similarity. So the transcriptomics fingerprint, basically, of exposure scenarios. And by grouping these chemicals, we can kind of do a read across. We can kind of match them in groups, which can then also feed into the AOP-Wiki by looking for all of the molecular initiating events that can be or that are described to cause a certain molecular initiating event, and therefore go more deeper into the AOP knowledge. So this opens many possibilities to make connections between the different case studies. And this will also be presented in the ISMB conference on a poster. So of course the project is close to finish in this November, but there's some ongoing work that is related to AOPLink. One section is the integration of the AOP database. AOP database is different from the AOP-Wiki. It is a tool developed by the US EPA, and they were winners in the first OpenRiskNet implementation challenge, which basically means that we will integrate their tool in the OpenRiskNet infrastructure. And on the figure on the right, we can see how this kind of fits in the AOP knowledge base, how they see the interaction. So they are very closely related to the AOP-Wiki, but contain many more pieces of information, many more external data sources connected to the adverse outcome pathways. They contain information. It contains information for chemicals, genes, pathways, molecular pathways, toxcast assays, and diseases. So extracting these, if I look at the results of the poll, extracting these diseases from the AOP database would be a really nice thing to have, to put this in a large Jupyter notebook or other workflow system. So this AOP database, at least the data that is underlying it, will be converted into RDF and will, similarly to the AOP-Wiki RDF, be exposed as a SPARQL endpoint. If you're interested in the AOP database as itself, it has been presented a few months ago by Holly Mortensen herself, also for OpenRiskNet, which you can find the recording for with a link below, or just find it on the OpenRiskNet website. The other section that we still want to add to the AOPLink case study is PathVisioRPC. PathVisioRPC is a tool to analyze, to perform pathway analysis based on gene expression data. And this PathVisioRPC allows to use the functionalities of PathVisio without needing to install the software, and is available for many of the preferred programming languages, including Python. So we can integrate it into our Python notebooks or R notebooks, Markdown notebooks, whatever is available. And we can use this tool to integrate the data, for example, that was made available and curated by DataCure, and highlight this data on one of the pathways that is available in WikiPathways to show the expression data, to visualize it, and to analyze it. And this would really be beneficial to make complete data analysis workflows of data inside of the OpenRiskNet project, inside the cloud, inside the OpenRiskNet infrastructure. And this brings me to the end of my webinar, of this webinar. Of course, my My work is funded by OpenRiskNet and also by EA-Toxrisk, so a shout out to EA-Toxrisk here as well. Thank you very much for listening all, and I'm open to questions. If there are no questions, then I would like to thank you all very much for listening in, and I hope that you have learned something useful, and hopefully we can answer all your AOP related questions in the future through Jupyter Notebooks. Thank you.